Hi class, let's talk about the spectrum of activity for our drugs. So are we going to be working with broad spectrum drugs or narrow spectrum drugs? To review very briefly, our broad spectrum drugs are going to affect many different kinds of bacteria, and our narrow spectrum drugs are going to have a very targeted specific group of bacteria or microorganisms. So if we think about this, our broad spectrum drugs are going to target something that's common to all bacteria, and our narrow spectrum drugs are only going to target a specific subtype or subgroup of bacteria, something that's going to be unique to a category or genus within our bacterial kingdoms. So if we look at our tetracycline-based antibiotics, generally speaking, those are going to be very broad spectrum drugs. They can affect many different kinds of bacteria, gram-negative, gram-positive, or gram-indeterminate, like our mycobacterium. If we look at our narrow spectrum drugs, our polymyxin, they're going to be much more focused, much more targeted in their destruction of bacteria. Class of drugs, the very first antibiotics are the penicillins. Our original penicillin was a very narrow spectrum drug, and it was very easy for the microbe to evolve or develop resistance to that penicillin. So it was very easy to have a counterattack mounted by the microbes. However, penicillin has been altered throughout the years. Through organic chemistry and chemical engineering, penicillin has been changed just a little bit. So now there are different kinds or different flavors of penicillin. And these different flavors of penicillin are better than the original one. They have wider, more broad spectrum activity, and it's more difficult for microbes to become resistant to our broad spectrum penicillins. So if we look at penicillin now, our penicillins can now affect gram-negative bacteria, they can affect gram-positive bacteria, and they can also affect our chlamydias as well. If we look at our isoizazids, those can typically only affect tuberculosis. Streptomycin can affect tuberculosis and our salmonellas, plagues, and gonorrheas, so our gram-negative bacterium. Our tetracyclines, as I mentioned, that are very broad spectrum, will affect rickettsias, chlamydias, gram-positives, gram-negatives. So some antibiotics can affect many different kinds of bacteria. Other antibiotics, like polymyxin, for example, have very targeted approach. So if you look at polymyxin right here, it can only affect gram-negative bacteria. So depending on what the diagnosis is, you need to prescribe the correct kind of antibiotic. You can't just give any antibiotic for any bacterial infection. Some antibiotics will not harm or destroy specific kinds of bacteria. So, what do I want you to know from this table? Not that much. I want you to focus on the name of the drug category. So we have our penicillins, our methicillins, our ampicillins, our amoxicillins, and I want you to focus on the spectrum of action. If you want to read about the uses, so what the molecular target is and what the disadvantage is, knock yourself out, but I'm trying to make this more approachable. This is an introductory microbiology course at all, not a pharmacology course. So I want you to focus on the name of the drug and if it's narrow or broad action. So penicillin G is narrow action, penicillin V is narrow action. Our methicillin, so we can see our cillins here. These are all the penicillin categories of drugs. Our methicillins are narrow action, but our ampicillin, a newer kind of penicillin, is broad action. If we look at these different kinds of penicillins, they all have the same beta-lactam ring, the same 4 and 5-sided rings that form the base of the penicillin molecule, or the cillin, and then these side groups are what have been changed through chemical engineering to make the different kinds of penicillins have narrow or broad actions. So our penicillin G, penicillin V, our methicillins all have narrow action, ampicillin, amoxicillin, and our mesolicillin, oh good gravy, that's a tongue twister right there, ticarcillin, those have very broad action. I'm sure I butchered that pronunciation. If we look at biofilms in particular, biofilms are going to have different species of bacteria working together and behaving differently. There are going to be different phenotypes expressed, or the bacteria are going to be in different phases, whether on the top or deep in the bottom of the biofilm. So because these biofilm bacteria behave differently, antibiotics are going to affect them differently. Many times, bacteria and biofilms will be unaffected by antimicrobials. That's a big problem. Another big problem we have is that thick slime capsule makes it very difficult for the antibiotics to penetrate that slime to get to the actual bacterial cell. And then finally, those bacteria in the biofilms are going to have different phases or different phenotypes expressed, which means that they're going to be susceptible to a different kind of antibiotic than what they normally would have been if they had been free-floating bacteria. So this makes the treatment of biofilms, once they become established, to be much more complicated and much more difficult for the attending physician. So, biofilm treatment strategies include one that didn't make the list, and I'm just going to throw out there, is don't get the biofilm. Don't get sick in the first place. Simple hygiene, such as washing the instruments and changing dressings, are a great way to treat biofilms. So a great treatment for biofilms is don't get the biofilm. But let's say you do have a biofilm that you need to treat. If that happens, then you can interrupt the quorum sensing pathways within the bacterium. Remember, quorum sensing involves the bacterium sensing the population density. And when a bacterium can sense a high population density, they start to act differently compared to bacterium that do not sense the high population densities, that only sense a low population density of bacterial cells. So if we can reduce the ability for the bacteria to sense that there's a high concentration of bacteria or bacterial neighbors, we're going to reduce the ability for the bacteria to form those biofilms. Daptomycin has shown success in the treatment of biofilms. Another approach to treating biofilms is to add a DNA. So to add an enzyme that destroys DNA. So if we add these DNases, we can destroy extracellular bacterial DNA, and we can start to hopefully penetrate some of the extracellular debris to get to the bacterial cells themselves. If we combine a DNase with an antibiotic that destroys or perforates the cell wall of the bacterium so that DNase can get into the bacterium, hopefully we'll have a synergistic effect, a 1-2 punch. And then finally, if an artificial device or implanted device is going to be put in a patient, it could be impregnated with an antibiotic, or such as silver, which is a very common one. Silver nitrate is impregnated in a bunch of stuff now, to make it more difficult for a biofilm to become established on that device. Some antibiotics are actually going to cause the biofilm to form at a higher rate than what they normally would because the bacterium act differently when exposed to the antibiotic. So instead of being an antibiotic, it's actually a probiotic, so to speak, in the sense that it helps the bacteria to grow. That was a really poor use of the word probiotic. I should say prebiotic. That's a technically correct term. Now, when we try to treat a fungal infection, we have some low-hanging fruit, and we also have some problems as well. A problem is that fungi, oh darn it, just happen to be a eukaryotic cell just like we are. So they present some unique problems for when we are trying to come up with a chemical that can treat them. Drugs that are designed to act on bacteria are very ineffective. They have almost no effect on fungal cells because the fungal cell machinery is very different from the bacterial cell machinery. The similarities that are between the fungal and human cells also mean that if the drug is toxic to the fungal cell, it's probably going to be toxic to the human cell. The fungal ribosomes are nearly identical to the human ribosomes. There is very, very, very little difference between the two. And because of the similarities between fungi and humans in terms of cellular machinery, there's going to be a very few number of antifungal drugs compared to the large number of antibiotic drugs. If we look at our antifungal drugs, they include our macropollines, our azoles, our echinocandins, and our nucleotide cysteine analogs. So what do these guys like to target? These guys are going to target fungal cell membranes. These guys can target sterol synthesis or hormone synthesis within our fungi. or we can target fungal cell wall in synthesis. Recall the fungal cell wall is made of chitin, and this is actually a structure. Oh, here we go. There's the mouse. This cell wall is present in fungi but not present in human cells. So this is going to be a nice target. These drugs should have a lower toxicity for human beings compared to drugs like our macropolicines that are going to target something that's present in both the fungal cell and the human cells. And then finally, if we look at our nucleotide cysteine analog drugs, these guys have a nice benefit of being able to be absorbed across the blood-brain barrier. The problem is that these guys are going to target both fungal and human cell structures. But I can't emphasize this enough. To have a drug that can go into the cerebral spinal fluid, so it can be dissolved across that blood-brain barrier, makes it a very powerful drug. Most drugs are not capable of being dissolved across the blood-brain barrier and need to be injected via a spinal tap. So, antifungal drugs are few and far between. Another one that's few and far between are anti-protozoal infections. Protozoa have eukaryotic cell structures. Quinone is the principal treatment of malaria, and it's been the principal treatment of malaria for a very long time. Quinone was used to treat malaria before we even knew what quinone was. It was initially a chemical that was present in a tea that was made, and that tea was found to help treat the presence of this malarial infection. Since then, we've now synthesized quinones in a laboratory setting so we can produce them at an industrial scale, and we've modified the quinone slightly. Now we have a quinine. We can now have our quinolones, our chloroquine, and our primaquine. These are all going to be like those multiple kinds of penicillin in that there's the same base functional groups of that molecule, and then there's different side chain functional groups to make them act slightly differently. Several different species of Plasmodium and many stages of its life cycle mean that this drug is no longer universally effective. Plasmodium is the genus of the organism that causes a malarial infection. And a big downside of these plasmodiums is that they have very, very, very high population counts, and they have very drastically different life cycles, which means that these plasmodium organisms are capable of developing resistance to the quinones relatively quickly. Just to give you some side history, the reason a gin and tonic is so popular in Britain or England is because when Britain was a world empire, and Britain was colonizing Africa and India, to fight off malarial infections, British troops would drink their quinone in a tonic. And to make that quinone more palatable, because it's incredibly bitter tasting, they would add some gin to make it more, to help it go down easier. And eventually they just got used to drinking gin and tonic. The thing that makes tonic, so those of you who like mixed drinks, the thing that makes tonic taste so bitter is actually quinones that are present in the tonic. Low concentrations of quinones, though. I should also emphasize that drinking tonic is not going to be a good anti-malarial treatment regimen. You'll need to actually take the tablet form with the quinones. Some other antiprotozoals include metronidazole, and this is a very widely used antiprotozoal that will affect amoeba, so it's an amoebicide. So if there is any kind of an intestinal infection, or giardia, or trichinomonas vaginalis, so if we have a vaginal tract infection, or if we have a digestive tract infection caused by some kind of an amoeba, which is a form of protozoa, then we're going to prescribe metronidazole. Other drugs that are going to have antiprotozoal activities include our quinacrines, our sulfonamides, and our tetracyclines. But those are limited antiprotozoal activities. Our two biggies are the quinine categories and the metronidazole category. I've got to practice saying those drug names. We also have anti-helminthic medications. There's a big problem, though, with helminthes. They are multicellular microorganisms. I'll say it again. They, generally speaking, are going to be multicellular. Sometimes they are microscopic, sometimes they're even macroscopic. And because of their multicellular nature, they have different tissues. They actually have tissue types now, and their physiology is going to be incredibly similar to human physiology. This means that we are going to have to come up with some novel treatment approaches. Some common helminthic infections include flukes, tapeworms, roundworms, and some other larger parasites as well. The physiology of these guys is so similar that it makes it difficult to target them. And just because we can block reproduction doesn't mean we're going to block damage. An adult helminthic worm can crawl around and continue to eat and cause damage even though it's not reproducing. So just blocking reproduction in and of itself isn't enough, because that adult worm can continue living in us as the human host throughout the course of the anti-helminthic therapy, and then when we're done taking the medication, start to reproduce again. So instead of targeting reproduction for these large multicellular organisms, we're going to instead focus on disintegrating or inhibiting the metabolism of the drug of the helminthic worm, or immobilizing the worm by paralyzing its muscles. The downside is these drugs have enormous side effects on human beings. So if we look at some drugs that we can actually use to treat helminthic infections, they include mebendazole and aldanazole, and those are going to inhibit microtubules of worms, eggs, and larvae. Microtubules are some of the structures that form the cytoskeleton. So by inhibiting microtubule formation in the worms, or the worm's eggs, or the worm's larva, we're going to cause the cells to become destabilized within these organisms and potentially lyse. The downside is there are going to be some pretty severe side effects for the human beings as well, because we as human beings also have microtubules in our cytoskeletons of our human cells. Crud. That's not going to be a very good medication to take then. There's going to be some awful side effects. We can also take the pyrantel. Pyrantel is going to paralyze the muscles of the intestinal roundworms. The downside is we also have a lot of muscles in our own bodies that can become paralyzed as well by taking this medication. So you definitely do not want to take this medication if you're doing any kind of athletic competition. We also have the praziquantel, and that's going to be particularly focused on tapeworm and fluke infections. We also have ivermectin, and that's going to be used for the treatment of strong lielodiaz and onchonoresis in human beings. This has been a veterinarian drug that was initially developed for animals. One of the nice things about veterinarian drugs and veterinarian science is there's fewer ethical considerations when doing testing on animals and developing a drug for an animal compared to developing a drug for a human being. So generally speaking, there's more rapid development and more rapid response with the drug production for animals. The downside is there's a heck of a lot less funding for the drug development for animals. Generally speaking, most people don't care as much about a horse or a cow or a dog as they would their mom or their dad or their sibling. So they aren't as willing to donate money for veterinarian drug development. So it kind of evens out in the end. It's easier to do, but there's less funding available. Ivermectin is an example of a drug that was developed initially for veterinarian use and now is made the shift to be used for human beings. We also have antivirals that can be used to treat a viral infection. Something that's unique, though, is that virons, particles, are not actual cells. So there are going to be fewer targets. Many viruses depend on the host cellular machinery for reproduction. So if we target the host cellular machinery, yes, we're going to hurt the host cell, but we'll also harm the reproducing viron particles. This disruption of the viral metabolism means that we inherently must disrupt the host cellular metabolism at the same time. The best therapeutic option for a viral infection is do not get the viral infection. Get the vaccine. So measles, mumps, hepatitis. Generally speaking, we have a don't get the disease approach. So get vaccinated. for AIDS, influenza, and the common cold. These are viral infections that have very high mutation rates, and we don't have effective vaccines for, and that we don't have effective medication for. These viral infections cause quite a bit of damage right now. Now, the common cold is just mostly an inconvenience. Influenza actually kills a significant number of people, particularly if you work with geriatric populations. Usually, there's a big increase in influenza deaths in nursing homes after Christmas time. It's, for whatever reason, this is very anecdotal. Most people I know that work in nursing homes will attest that a lot of their patients will like to hang on for one more Christmas season, and then after that one last Christmas season, there's generally speaking is a lot of influenza deaths in the nursing home situations. And then AIDS doesn't really need any explanation, but if you do need more of an explanation on AIDS, we will get that in a later chapter when we talk about diseases of the immune system. So what are our antiviral medications going to focus on? We can focus on keeping the virus out of the host cell. If the virus can never get into the host cell, the virus can never take over the host cell and then have reproduction. So this is a preferred mechanism because it's going to cause minimal damage to the host cell metabolism. We're going to focus on keeping the virus out of the cell. That's an excellent one. We also have blocking transcription and translation of viral molecules. This, generally speaking, is going to target host cell metabolism. So there's going to be some nasty side effects for those antiviral medications. And then finally, we have the prevention of the maturation of the viral particles. This, again, happens in the host cell. So we're looking at disrupting host cell metabolism. So dang, this approach is also going to have some strong side effects, some unwanted side effects. We also could look at some DRACOs. DRACO stands for double-stranded RNA-activated capsase oligomer. The caspase cascade is a metabolic pathway that's activated when cells are self-destructing or going through the process of apoptosis. So if we can deliver a DRACO, double-stranded RNA-activated caspase oligomer, we can initiate self-destruction of cells that are infected with double-stranded RNA-based viruses. So if that virus stores its genetic information as double-stranded RNA molecules, this drug can initiate the caspase cascade and cause that cell to self-destruct. So any cell infected by the virus will immediately destroy itself before the virus can reproduce. This is currently the new kid on the block, so to speak. It's not very well understood, and some drugs are currently in development based off of this mechanism. So, let's look at some antiviral drugs you can actually get on the market right now. If we look at, and let's focus on these formal names, although you would probably know them more by the brand names. We have enfurotides, our amantolidides, and our zanamivers. These guys, and also our oseltamivers. These guys focus on blocking viral entry. We also can focus on inhibiting nucleic acid synthesis. So if we can't replicate the viral genome, the virus can't reproduce. So we can have a cyclovir. That's a very common one. I know many people who've had shingles have been given a cyclovir. We also can have rifampin, or we can have I'm not even going to try to say it. So here's a big hint or clue for those of you that are watching the video. If your instructor can't even say the name of of this molecule that makes up that antiviral drug, he's probably not going to test you on it. But what I do want you to know for the exam is that viruses or antivirals will focus on blocking viral entry or nucleic acid synthesis or the release of the viral molecule. There's a big hint for your exam right there. So that's what you should focus on from this slide. So, concept check class. We have a bunch of drugs listed below. Which ones are antimicrobial, antifungal, antiprotozoal, anti-helminthic, and antiviral? This one is a doozy of a concept check question. Take some time, flip back through your PowerPoints, flip back through your textbook. It'll probably take you about 5 minutes to look up the answers to this question. Or you might just punt and do a Google search. So pause the video, get me some answers. 1, 2, 3, 4, 5. All right. Ribavirin is an antiviral drug. A big clue right there is that virin name. So if you see a virin within the name of a drug, it's going to be an antiviral. If you see a cillin, that's in the name of a drug, that's going to mean it's a penicillin derivative, so it's going to be an antibiotic drug. Metronidazole is an antiprotozoal. Mebedazoles is an anti-helminthic, and then amphotericin is an antifungal drug. If you have any question about the spectrum of activity of our antimicrobial drugs, please feel free to post them in the discussion board or shoot me an email. Happy studies!